Okay. Good afternoon, everyone. My name is Orian Tzadik. I'm coming to you formally from the University of South Florida, currently from the Pew Charitable Trusts. And today I'm going to talk to you about a paper I published in LNO Methods a couple years ago. It's entitled Chemical Archives in Fishes Beyond Otoliths. And this is a review that we did on the use of alternate body parts to otoliths as chronological recorders and microchemical analyses. Before I begin, I definitely want to thank the ASLO board and everybody here for coming to listen. And considering this is an author spotlight session, I definitely want to spotlight the other authors on this. This was a collaborative work. This was something that was done across several labs at USF, and they're continuing to do really good work there. Continuing with the theme of the author spotlight, I'll tell you a little bit about how this came to be. So I started my PhD at the University of South Florida in 2011 and almost immediately got involved with a project on Goliath grouper population assessments. Goliath grouper is this grouper you see right here. It's the largest grouper in the Atlantic and the second largest in the world. They get up to 8 feet long historically and up to 800 pounds. And so they were the target of a lot of fishing, and they've been fished down to a point that they are now considered critically endangered. So we're doing a population assessment, and we wanted to find out, we had a lot of questions about diet and movement, fecundity, things like that, but we wanted to look at these things non-lethally. So we had to get a little bit creative. So normally, if you're looking at diet of a fish, you would cut open the fish's stomach and you'd see what it was eating. If we don't want to kill the fish, you can use an alternate technique of sticking your hand down their throat, pulling out what they've got in their stomach, putting it in a bag and analyzing it. So we did a lot of that. The part that I was involved in, well, I was obviously involved in that, but my research focused on the fin rays. So what we do was we would cut out 2 soft dorsal fin rays, like that, and we would take them back to the lab and we could analyze them to look at age and growth. My study then came in and said, maybe we can do more. Right? We've seen in otoliths we can do a lot of chemical analyses, so maybe we can do the same thing in fin rays. And that's what I did. And we looked at diet and movement and habitat use across fin rays, using the fin rays as proxies. The best part about using fin rays is that they grow back. And this all led to then collaboration among some of these other labs at USF, where we started to think about, well, what other structures can we use on fish? What other things can we do, can we look at as proxies? And that led to the publication that I'm talking about today. Now, I put this picture up just because I think it's really cool. A bit of a nerd, sorry, but what you're looking at, it's all the same picture. These two are this little box blown up, and what you're looking at is a bone in transition in a zebrafish. So Julia Mohammed did her PhD on this work, and she watched zebrafish, how they grow and how they uptake elements. And so I think a lot of us in this room know that when you uptake elements into your body, it's through either diet or through the ambient environment. With fish, what that means is that whatever they're eating or whatever they're swimming through, they will then use to put into their structures, and in this case, bone. And so you can see here these little, well, they're better here, but these little kind of globules, and they are mineral deposits that are getting transitioned into hard bone here. And so the minerals go into the bloodstream, they form platelets, and then they form plates, and then those layer on top of each other to form bone. And so again, I just like the picture, that's why I put it up here. Okay, the gold standard for a lot of this has been Otolith Science, right? We have a calcium carbonate little rock inside your head that works to help with balance, and it's comprised of calcium carbonate, like I said, and it's a aragonite structure. And it's largely inorganic, almost entirely inorganic, and it has a continuous growth throughout the life of the fish. It's very clear to read. You can see up here, right, the growth bands. There's a very distinct annuli, and in a lot of species, there's very distinct daily rings. So you can do age and growth reconstructions with otoliths, and like the way I was just talking about, you can do microchemical analyses and microstructure analyses. And there's also a long record of these samples because people have been looking at them for a long time. In addition, because they're inert and because they encapsulate well, archived otoliths, like skeletons in a museum, can often be used as well. And so all these things combine to say that a lot of people have called the otolith an ideal material. However, there's one major drawback to otoliths, and it's that that's how you have to extract them. You have to rip open the fish's head to take out the otolith. So obviously in a situation where you've got an endangered species, that's not ideal. And if you work with Goliath grouper, then you have to wait until you get a nice, big, fat, rotting carcass on a beach, and you get called to go on a Saturday morning and slice it open. And that's me using a power saw to get into it. You can just imagine the rotten fish with the power saw, and you know, it gets a little stinky. It's not fun. So there are a couple drawbacks to otoliths, right? Major one being, like I just pointed out, it's lethal sampling. This creates a problem with endangered species or threatened species. It also prevents you from doing any future monitoring on the fish. You can't rip open the head twice. It's also largely inorganic, which is great when it encapsulates previous layers, but not so great when you want to look at a proteinaceous matrix. And so if you're interested at all in the protein, you should look to other structures potentially. There's also a problem with vateritic otoliths, which is just fish under stress sometimes, instead of creating the aragonite structure, will create a vaterite structure, and that becomes very difficult to read and much more difficult to do any sort of chemical analyses. So that's enough background. Let's get back to the paper. The alternate structures that we talked about in the paper were the endoskeleton, here exemplified by the clythrum and operculum, scales, fin rays, fin spines, eyelens. Quickly, fin spines have bones. They're the bony hard ones. Fin rays are the kind of cartilaginous ones, but they're actually very similar in structure, just a little bit different in the amount of calcification. I hate putting tables in talks. I'm only putting this here to tell you that it's in the paper and you should look at it. The paper is about these different structures, but it's also meant as like just a reference. And so this table in particular is that quick reference, right? If you want to look at endoskeleton, can I use it for aging? Well, yes, but you need a species-specific understanding of bone remodeling, etc. So I thought I would quickly run through the different species, pointing out a couple structures, pointing out a few advantages and disadvantages of each. And so when you're looking at endoskeleton, like I just pointed out, it's all very species specific. And so different bones work differently with different species. It's a little complicated, but often you can use it for aging. However, obviously this requires some lethal sampling, but there's also problems with bone remodeling. Bone is a resilient structure that likes to replace itself. And so new material will replace old material, and that's not ideal when you're trying to look at a chronology of things. There's also species-specific turnover rates of the collagen within the bone, again, pointing to problems with chronology. All right, so then fin spines are potentially non-lethally sampled, and we've seen studies that have looked at movement patterns via trace element analysis. It has a few limitations, namely vascularization and resorption, which is a fancy way of saying sometimes that middle bit gets eaten out and you can't really see those first annuli. There's also a bit of a problem in a lot of these structures, as was mentioned before, with edge crowding. And so once the animal gets much older, it doesn't grow as much, and so those bands are much closer together at the edge. That's particularly problematic with something like a scale, but it's definitely problematic even with an otolith or a fin spine or anything like that. There is a proteinaceous matrix in fin spines, but the utility is yet to be tested, to my knowledge. Fin rays, on the other hand, are something I'm a lot more familiar with, and these have the advantage of being non-lethal. Like I said, they grow back. You can look at both movement and diet studies via both trace element analysis and stable isotope analysis. So there's a nice proteinaceous matrix as well as a mineral matrix in a fin ray. Again, the limitations are going to be the same as the fin spine with the vascularization and resorption. Scales have a great advantage in that they're very easy to sample and very non-lethal. You can kind of just rub the fish sometimes and the scales fly off. And again, there is potential for movement in diet studies via both trace element analysis and stable isotope analysis due to both the proteinaceous matrix and the mineral matrix. There are some limitations with scales, including edge crowding, metabolic reworking, overplating, and inconsistent circuli formation, which is what I was just talking about. The last structure is an interesting one, a little bit newer to the scientific world, and that's eyeballs. So eyeballs kind of grow like onions, and they have the lenses go on top of each other like that. Under a dissection microscope and very fine tweezers, you can actually just peel off the lenses and put them in a little tin capsule and then put them in your stable isotope. There is also potential to look at trace elements with eyeballs. It's been done rather not terribly successfully so far, but the point is that inner layers retain their chemical makeup, and so you can look at either proteins or minerals over time with these. And as I point out here, it's a very easy workup. It's just, peeling an onion, really. The limitations are obviously lethal sampling, and the suitability for aging studies is unclear. We're pretty confident that they do go in a certain pattern and that there's, let's say, 4 lenses a year or something, but it hasn't been worked up, to my knowledge, on any species. They're also completely uncalcified, so that has its own drawbacks. All right, so at the end of this paper, we identified some future research, some gaps and things. What else can we do moving on from here? And I'm just going to run through those quickly. We think it would be really important to combine structures and perform both trace element analysis and stable isotope analysis, see about the interaction between them, what's going on at the same time. Ideally, if you could just run on an ICP-MS a laser that goes down and get information about the proteinaceous matrix as well, that'd be amazing. There's a lot of species-specific stuff that we don't understand, and that'll be these next 2 bullets. Also, I want to point out alternate analytes. So we don't currently really look at anions at all, even though that could be a really interesting path of research. The reason behind that is just because it's really hard to do, and so nobody's done it yet, but there remains a lot of potential on that. Also, future research on elemental uptake in different environments and creating kind of background information element maps and isoscapes, as well as looking at different alternate structures, continuing to do that. I'm running a little low on time, so I'm just going to say at the end here that this has drawn a lot of interest, this field. It's very exciting. This paper has drawn a lot of interest that I think is great. I'm going to tell you a really quick story about a researcher in Iran who wrote me an email and asked for a copy of the paper. He said his government blocked the paper for whatever reason. And he's actually stayed in touch, and he sends me Christmas cards and all the rest of it. So it's pretty cool to see that this kind of thing is taking off. Again, just this is about more information on further where this has been presented and received. And these were some of the next steps that came out of the International Otolith Symposium and AFS last year. I just want to point out basically adapting these alternate structures for use in management. I think that's exceptionally important and something that people in this next chart, what I'm showing, people have a lot of optimism when you create a proxy, right? They say strontium isotopes can pinpoint geographic origin. Then you kind of get to this pessimism phase and you get this, well, it's overused, there's too much confidence in it. And then you get to the realism phase, which is strontium isotopes can be a powerful tool, right, to estimate geographic origin. So in a similar vein, I think that's where we are with a lot of these topics. And unfortunately, I think a lot of the time we're in this pessimistic phase. And I think that we need to move into the realism phase to know what we can actually do with these structures and how we can adapt them to management. And with that, I'll say thank you all for listening.